Hi, Clementine.
Hello, how are you?
Good, how are you? Congratulations, Dr. Burrell-Sander.
Yes. I mean, annoyingly, technically, I can't use the term doctor until June
Why?
because basically the university has to process my like thesis, whatever else. And if I had done it a day earlier, it would be
So I
processed by
cannot hear you
March.
anymore.
Oh, that's weird. Can you hear me now?
Oh, why?
Hello? Oh, okay. So it's not just you. All
Let's
right.
try
Hello.
now.
Okay, hello. We're back. But yeah, basically, I missed the deadline by a few days and they process
Mm-hmm.
them in batches. So the university won't process it until June unless I ask them to do it early for some reason.
But whatever is 6 more months, right? Or 4 more months.
Yeah, and I mean, if I really cared enough, I could ask
Mhm.
them to process it. But, for now, I'll just, I don't think they're going to stop me writing doctor on my emails.
Yeah,
You
of
know,
course. You deserve it.
Interesting thing about Australian PhDs, right, is there's no Viva exam.
so there are now. I just did my
Really?
Viva last week. UCIT is now
Oh,
doing it.
really?
They've made it optional to do a Viva. And, well, I mean, um, when Phil Gale was head of school, apparently he wanted everybody to, he wanted to make it mandatory for everyone to do a Viva. Um,
Mm.
and I mean, basically, Peter has not enforced that now that he's head.
Right.
Um, I think it's just down to the supervisor. And I wanted to do one so that I would have the experience of doing a Viva. Um, so I did.
Yeah, great. Well, it's from an examiner's point of view, it's very interesting because the Viva is really appealing because you then get to interact with the candidate and you, in one neat movement, overcome the AI problem.
Yes, yeah, we were slightly worried because I'm in the UK. Peter was slightly worried that they would say that it doesn't count because it's on Zoom. But I think they
Mm.
hadn't thought about then it didn't occur to them to question it being on Zoom because we now
Right,
our Vivas for honors students are now in person only because of
right.
AI concerns. Um, but yeah, I mean, I was a bit torn about whether to do it because it's essentially more work because it's optional. But it
Mhm.
is kind of much nicer to explain things in person rather than to have to write oh yeah, I
Mm.
see how you got confused, but actually this, like,
Mhm. Yeah, yeah, for sure.
and I mean, I've heard some terrible stories about people's vivas from British, like, people who've done it in the British system. My
Mm.
examiners were very lovely. So
Okay. I mean, the reason for having two and the reason for having an internal is the internal is meant to be the one who makes sure that everything is fair. Um,
yeah, so
that's the idea.
I had the two examiners who read my thesis anyway, and then a we have an internal, what's the word? She's not an examiner, she's just like the chair. So,
Hmm.
theoretically I
Hmm.
could have gone to her and said that they were mean
Yeah,
to me.
right.
But fortunately no need.
Great. So it went well. That's great.
Yeah, the
Wendy is
worst part was that,
sitting. Oh,
sorry.
Wendy is sitting next to me today,
Ah,
Oh,
hello.
okay.
and there's also Saskia and Deborah.
Okay.
All right. I
Yeah, I
should
was
make a
just
start.
yeah. Oh yeah,
Okay.
Peter and Erica won't be coming.
I think Erica is teaching. Yeah. and Peter is teaching.
Yeah, they both said they were teaching classes in person, so
Okay, let me share. If I could find, okay. Okay. Right. Well, welcome everybody. Hopefully that is clear enough. Okay, so I mean, we might as well just start going through this, I guess. The LinkedIn page, the fact that's up and running is really great. So Max presumably is the admin, right? Is that right?
Yes, it's Max and me.
Okay, great. So so when I brought it up on my browser and posted something, and I think I posted it as, as MycetOS or something.
Yes, yeah, I saw
And
it.
then right, and then I tried to comment on something or like it. And I think I also liked it from there, so I had to switch to myself. I guess if people are going to put things on the page, they've just got to be careful about who they are when they're doing that. So I mean, we can post as ourselves, I think, or post as the organization, I guess.
Yeah, so everyone who clicks the link can post on the MycetOS page. So it's indeed that we're trusting everyone who can click to post something relevant and not something bad. So there are like 2 or 3 posts right now on the MycetOS page, LinkedIn page. And for the next, I already asked Clementine if she wanted to put a picture or her pictures from the past 4 years working for MycetOS, since she officially graduated now, like working in the lab with students, for example, or when she was here at Erasmus, to use a picture like that as the next post. So it's mainly, I do get updates on how the MycetOS page is going, the impressions it gets, and how many people started following it. So it is growing, but we need to keep making posts and keep liking it as ourselves to spread it more.
Did you say that anybody can post to it?
Yeah, I was surprised that you could post. I thought it was only me and Max, but I think
I thought that he gave people, you two have admin privileges, and then he gave a lot of us posting privileges. So it's not everyone.
Yeah.
Yes.
Yeah. Oh yeah, everyone, a few of you as posting.
Okay. So I mean, I guess the one way to grow it well is to tag people, right? So to tag people from WHO and people from the CDC working group and all these people, to try to get them to come and follow it. And we just gradually work. I mean, that's how we grew the Twitter following, was doing that.
Yeah.
And we can also, of course, for the next monthly meeting, a week beforehand, try and post something there that highlights the fact we're going to meet up again. Those are good for alerts to the meetings coming up.
Yeah.
Yes.
And Karen.
Sorry, I started trying to just put together a basic kind of essentially template so that we could have a bit of uniformity, but also I can add to that just next meeting is X time, here's the link, etc. And then all we have to do is just make sure the dates are right and just post it every month. Yeah.
Okay.
We also talked about I think you weren't here last meeting. We talked about having talking about papers that we've published, saying like, oh, did you see our paper from last year, whatever, and PhD students, honors students joining the groups or submitting, etc. But then also maybe to get it started, doing a kind of like meet the team series. So it might be, here's 4 people from UCID who are working on it, and we'll post about them. Here's people from UCL, here's Erasmus, etc. So I've got some kind of draft templates for that, but also if people would rather just throw it together themselves, they can do that. But I was trying to make something where the idea is that, some people can just answer a couple of quick questions and say, oh, this is what I do, this is why I joined. And then one person can put it all together and press publish.
Yeah, okay. Sure. Yep. And also, I mean, yeah, I mean, people do tend to watch a lot of short form video content, right, as well. And so, you know, if someone is doing a poster at a conference or if anything is coming out like that, in addition to the idea of a static photo or a post or a link to something, we can also do, you know, 20-second video things as well. I know they're much more difficult, well, nerve-wracking in case you do something, you post something which is ill-advised, but you know, these days on LinkedIn, there are so many little videos of people talking about things. We shouldn't neglect that too.
I'm
All right.
gonna think about that and to see if I can come up with a fun video about something.
Okay. Yeah,
Hey,
great.
you could even just do like, here's my poster that I
Maybe in the
presented
lab.
at where did you go? Brazil. And
Yeah,
just
I only have pictures of that and not really videos.
but even if you just, do you still have the poster?
Yeah,
If you
I
stand in
still
front of
have
the poster, get Max to film you.
Yeah.
I also still have the lanyards from the Congress.
All right, cool. And then the Who's Who page was the other development, and I think you did that, Clementine, right?
Yes, I did. Just one more thing on the LinkedIn. Do we have
Yeah.
a specific page where we're talking about these things? I can't remember if Max made one, as in on GitHub.
I don't think so, but Max is not here
Yeah.
today.
And maybe if you can ask him and find out,
Yeah.
but otherwise we'll make one poster.
Okay. All right. That's good. But yeah, so people here should feel free to use the LinkedIn page obviously to post things that are relevant. And like I said, it can, just because we are a science organization, it can help always try and link something back to something concrete about the science. So if, I mean, if for example, yeah, you do a video about a poster, if you're able to post the poster, that's good. If there's something related to the meeting, post back here. If there's something related to a new request in the community or some development, being able to post back to an issue that discusses it, that kind of thing. So we try and make sure that the social media activity is grounded in something concrete. That always helps, I think. All right, series 2, I guess, the aminothiazole. So as I understand it, and I'm sorry if I missed something in the last meeting, synthesis is now complete of all the molecules that we're going to include in this in the paper, and the testing is complete of all compounds. There were some mentioned from Peter, and I don't know if those have been done. Anyone want to update?
I'll let Dunya talk about any testing updates, but yeah, as far as I know, all the synthesis is complete from UCID, definitely from where is it, the Uni of York. I think they had completed them, they maybe just hadn't sent them yet. So I think perhaps that's what that's why that was a bit unclear. And then I think Erica posted that they were trying to make some compounds with Whitgift that didn't work out, so we just won't be including those, which is fine in terms of our SAR and so on.
Right.
And yeah, I have a draft. It's just that Peter and Alice wanted to look at it before I shared it, and they just haven't gotten around to it yet, but hopefully they will soon.
Okay. Is there, do you want anyone to check anything else like the experimental data or something? I just wonder if there are other things that we could be doing while we're waiting that are going to need to be done.
Yeah,
or
that's
is it all
actually
in one file?
that's a good point. So I was hoping that the biology team would look through all the biology stuff that I've written.
Yeah.
And I can probably share that part right away. I don't think Peter
Right.
have an issue with that. And then the experimental, yeah, that would, and it would be good to just have somebody look through it in terms of proofreading. I've basically written it as, what's the word? There are sometimes compounds that I have more data for, but we generally have 3 data points for every single compound. So I've usually just written the 3 data points, if that makes sense, rather than have like full IR data for these 23 and not for these ones. And I've written it so that we have the thiazole intermediates and then the final compounds, rather than having the 2 steps of intermediates where you've got the benzoyl thiourea and then the actual thiazole, thiourea that we want, because that just makes it easier to write up. But yeah, I can definitely share the experimental sometime this week.
Yeah, sure. I mean, we can just be checking on the big things in case anything's missing. I'm not expecting anything, but just thinking typically speaking, the rule of thumb we have in my lab is that if you've got compounds which are new to the literature, then you need more data to justify that you can say you have made them. And anything like that, you would expect maybe the proton NMR or LCMS trace or something like that to demonstrate that they are pure. If those are available, that's great. If they're not, then usually any molecules that are of particular interest because of their biological potency, you would provide some evidence that they are correct and pure.
So generally we have proton and carbon for every single compound, without fail. And then for compounds that we've made at UCID, we usually have high-res mass spec data. And I mean, we've run them on the HPLC. I don't have a very nicely formatted trace, but we have that because that's how we assess their purity. And the only trouble is that there's a little bit of inconsistency between. I'm not quite sure what Peter was doing at the Uni of York, for instance. I think I don't think they were doing LCMS there.
Yeah, that kind of thing. I mean, if you've got a proton and a carbon, you don't really need the LCMS very much. I mean, the assumption is you haven't got it, full of sodium chloride or whatever. But if that document, the thing is, formatting that document so it's ready to go. And I guess the SI file that we had for Naramol paper that went to RSC Medchem is a good template. I mean, it's just really simple. So if it looks anything like that, we're good to go.
So that's the Naramol paper. Yeah, I'll have a look at that.
I mean, we can just, I think it's a good journal. It was well worth read. It was reasonably quickly done, and it's the right audience. And we just submitted it in that style. And so, that SI file we'll have a lot in there already, actually, that we need. But the template for how to present the information and how to present the NMR data is just the same, right? And it works
that's really helpful.
Yeah, and it should be on the journal as the Word file. I think if it's not, I can send that out or put it somewhere.
Yeah, I'll have a look. I mean, I'm very keen to get this paper submitted. So
yeah,
and also good for consistency.
Yeah.
So same group, same type of manuscript, same supplementary data.
Yeah, I mean, it's more that you don't want to. We know that format is acceptable for the journal, right? So there's no, then I'll come back and say, can you redo this file? All right, cool. And then if you're happy to put that file somewhere, or in some way share it, then the chemists here can have a look at it and offer comments. For the biology, do we know if the testing is complete?
So Deborah has taken over the in vitro testing. She's sitting there. And she said the last batch of compounds sent by Peter did not have any potential hits. I will put the results this week in the big spreadsheet.
Okay.
In that case, I will incorporate that into the paper as well.
Is that surprising? Did we have any molecules in there that we thought would hit?
From memory, what Dunya, like the compounds that she's talking about, were some of the variations where the N, like the aminothiazolamine was alkylated. So we were just curious about that. And then I think we had the pyridine, like making it with a 3-pyridine or a 4-pyridine instead of a 2-pyridine. And it's
okay.
not exactly surprising, but it's interesting that it's so, fussy about the position of that N.
Right. Okay, all good. Great. How many compounds do you think in the paper, roughly, do we have testing data for? How big is this thing?
Actually, that's a good question because I don't have it right in front of me, but the way I've written it up, I essentially didn't really talk about a lot of the compounds which are kind of irrelevant, as in we, to go back to the pyridines, we found out that the pyridine ring was necessary, and we technically have another 30 compounds that didn't have the pyridine ring because we were obviously hoping to, we didn't know the pyridine was necessary at that point. But I haven't written about that because it just seemed, more confusing. So if we look at every compound we've tested, we have over 100, like around 120, 130. But I think in the paper, I've included about 70 to 80.
Okay, no, that's great. In the supporting information, we have the table of everything?
So I don't have it specifically written for the paper. I have something very similar for my thesis. So it shouldn't be too hard to kind of, transfer it across. But yeah, that's one of the main things that I was hoping to get feedback, both from Peter and from the MycetoS group generally. Because I think it would be nice to emphasize that we made lots of these compounds, but we don't want to make it deeply confusing.
No, I think that's right. So, I mean, in the paper, if you have, a kind of matched pair, which demonstrates that some part of the structure is important, and then there are another 10 compounds with that same thing, and they're not particularly interesting, then featuring those in the paper, it doesn't really make a lot of sense. But listing them in a table in the supporting information would be useful, just because then there is the one set of everything. Yeah,
so I've got the, the smiles and the MyOS codes for everything we've tested, but I haven't put together any experimental data for the ones which are not discussed in the paper.
Right.
But I agree with Matt. I think it's good to include those so that at least they are somewhere published. And not only on the GitHub page, because people might see it easier there than on GitHub.
Yeah, so you've got characterization data for those, have you?
So most of them are made by UCL. I mean, there's obviously a few from us. So I don't think, because I've asked Erica specifically for the data for the ones which were going in the paper. So I know she's got the data somewhere, but I don't have it, saved on my computer.
Right. Because I think that if we are, it's nice to, if they can be featured in the SI, it is good,
to be honest,
I
haven't looked through the literature at those ones, but I suspect a lot of those are not novel compounds because in general, the ones which are made with phenols are more likely to have been made before. So in that case, it's presumably a little bit less crucial that we have lots of characterization data for them. And I believe Erica definitely has, like I said before, carbon and proton for them. So,
right. Yeah, look, so if there are molecules which are kind of inactive and not terribly interesting, and which have been made and characterized in literature before, and if we're able to say that our data match those, then you can get away with saying that these were made according to literature procedures. but I think it's important to try to make sure that the experimental section is complete. Even for the compounds that we don't necessarily feature in the main paper, just because then at least because there's no other way in which any of this is ever going to be published unless we put it in this paper.
Yeah, and that's what I was thinking about too. And I can talk to Erica and Peter, I guess, as well, Peter Rayner, to see if they can send me that data or if they've already got it put together somewhere. But yeah, like I said, I can tidy up a table so that you can at least see these ones are all written up, these ones were inactive, but we have the data or we don't, etc.
Yeah. Okay. All right. Very good. So so that's everything for the paper, I think, right? For series 2.
For the time being, I think. I'll try and post some things, things I've already got done. I'll try and post those probably by the end of the week. And then I'm not quite sure for the draft itself. But hopefully before next meeting.
All right. Okay. All right, great. For series 5, I think we're just waiting on some final biological data so that we could finish off the diagrams, and then the conclusions from the whole study. How is that going? Was that
do you they were only testing the final compounds against other strains, and it's still ongoing.
Okay.
It's currently being done. So once the results are in, I will put them in the spreadsheet.
Okay, great. Because the
duplicates but not triplicates of those.
We have doubles but not triplicates.
Yeah, because I think the reason why this was why it's reliant on that was because we needed to make sure that we were just including the right numbers in the diagrams for those averages. That's what it was, right? So we had to get those data before we could finish that off.
And there was just 1 or 2 we needed to repeat, and since the other ones came in, so we decided then to add those 2, so that everything could be in the paper.
Right. Hopefully, this link to the Series 5 manuscript clarifies everything that is remaining. But if there's any doubt, then we can talk about it. But I think it was basically stall on not just on that.
Yeah, we thought that when we finished the data, we went back to the manuscript and see if now everything is complete.
Yeah, hopefully. Okay, great. But so when we have that, I'm very happy to resume the writing part and finishing that off.
Yeah. Okay.
Just quickly, sorry, I realized I've forgotten something about the aminothiazoles. Clementine, for that paper, did you have a go at talking about potential mechanism of action in there?
Yes. So, basically, the crux of it is we would like to know about mechanism of action, but we still don't know too much.
Yeah.
So I talk about the fact that the CYP51 enzyme is kind of the one that we know best, and we don't know if these compounds act on it or not. And there is some testing that we could do. We could do some modeling. We could do some synergistic tests, etc. But it wouldn't necessarily be completely conclusive. And then what are the other 2 things? I briefly mentioned the calcium channels that we talked about it was probably 6 months ago now, where essentially the aminothiazoles have been found to block a specific calcium channel, the KCA 2.1 or whatever. And so I synthesized another separate compound that also blocks those same channels to see if it would also have activity against mycetoma. And annoyingly, it's inconclusive because the compound that I made is much less potent than the aminothiazoles. So it did have a little bit of activity against mycetoma, which, if it was working through the same mechanism, would make sense because, you'd expect it to work a little bit, but not be very potent. ideally, I'd go and make a couple of the more potent, inhibitors of that channel, so that we could compare them more directly. But all of the other more potent ones were more annoying molecules. There's like the cyclic peptide and these weird deliquin, I don't know, big ugly molecules, basically. So
okay. All right, fine. I think it's just important we address it, but we don't have to solve it.
yeah, and yeah, I also have a reference, well, not a reference, I briefly talk about the fact that the aminothiazoles have been explored as tuberculosis anti-tubercular agents, and essentially there's a bit of what's the word, like hypothesis, into how they might work on that, which could be relevant, but there's not enough data for us to say it is.
Okay. All right, great. Okay, series 7. So I'm a little bit behind the times on this. Does anybody want to chip in on current activity in this series in terms of molecules incoming and also biological evaluation? Lots of little bits going on here, and I'm not sure
So for
what's
the Notre Dame compounds, 10 of those that came in were potent. They're being tested, the MIC is being determined for those 10 compounds, which is also ongoing, but I will update once they're finished. Oh, and I ordered compounds in December. On Friday, one of them came in. So I'm still waiting for 3 or 4 compounds to come in.
Okay, so we're still waiting on a few. Is there a reason? Are they weird compounds, or is it just slow
No,
supply?
we never. So Medchem Express, sometimes it comes in 2 weeks and sometimes it takes months. So I ordered, I think, 1 compound from Medchem, and we usually don't order from Tokyo Chemical Institute or something. And
Right.
that was I think I ordered the bulk there. So yeah.
Okay.
But
Those
Kevin did give a call to follow up on where they are.
Okay.
compounds have been added to the master list, but not the SAR diagram.
Is the master list up to date on evaluation?
Dunya, did you add the high-low screening data? Was that
Yeah,
in the
for the
past
Notre
couple of
Dame, I
weeks?
added that. Yeah,
Yeah.
they're
So
already in there.
I think I updated the SAR diagrams right after our last meeting, so the end of January. So anything that's been put into the master list since then isn't in the SAR diagrams yet, I think. But yeah, I tidied up the SAR diagrams, and I kind of split them into some different categories, which has a bit of duplication across it because the set of compounds that we have are a bit, weird. And yeah, I'll have a look at and see what the results are and if that gives us any particular leads. But in terms of synthesis, I don't think Shane's not here today, is he? No.
No.
Yeah, Shane's definitely going to be making some compounds, but not until August.
Yeah.
And I think Peter posted about what he's going to make just on this page, on the
On the meeting page.
Right. Okay, I was just looking at the new diagram. So you split this up into separate bits.
Yeah, so that's what I mean.
Right.
There's duplication. I think like the lead compounds are in all 3 of them. But I was trying to split them up so that we can see kind of different trends to focus on. So I think that one that you're kind of on at the moment has the compounds that Shane made with UCID students, where you can see that having the methoxy instead of the free alcohol wrecks the activity. And then the benzimidazole are their own problem in terms of trying to compare them. Yeah, I
Maybe
don't
that's
know if
not
we
too
have
bad
any.
that the toxic group is inactive, because that might open a door to some product strategies, maybe ester compounds, suitable ester compounds, which reduce activity and toxicity of the compounds, but might be cleaved in the organism by the time and get their activity back to achieve a good in vivo activity in larvae or in mice or vertebrates.
Yeah, that's a very good point.
Yeah,
I
I
think there
think
are some examples, antibiotic examples of niclosamide, which were esterified with amino acids or phosphate, even phosphate, to increase water solubility and to reduce side effects.
I took a look at the lab notebook of Peter Rayner's group, and it looks like they're trying to couple aspirin to various anilines. So that might be something.
Yeah, I mean, now that I've seen what Peter is planning to make with his students, I can add those into those diagrams, as in progress, um, so we can have a better handle on what we're exploring or not exploring. Yeah, I think Shane was planning to stick to a variation of what he did last year, but that might have changed.
So it seems like, yeah, Shane is planning on the synthesis of specific new compounds, but only later in the year. And Peter, I think, made a comment that he was trying to work with students to improve the methodology, was it? So that
Yes.
the synthetic approach could be simpler or more suitable for more junior students, for example, with some of the schools we've been working with, I think. Is that where we are currently up to? Because I know that Peter posted some target structures, but it felt like he was more interested in the methods. Am I
Yeah, I
right?
looked at their lab notebooks. It looks like they're trying to use DCC to couple the two parts together.
Yeah, I mean there are 100 different ways of making amides, so I guess they're just seeking something which is as straightforward as possible.
Yeah, that was definitely the idea that Peter would try making some compounds, but figuring out the best routes, and then Shane would be able to use what they've figured out
Mm,
later in the
okay.
He
year.
reported, he also reported that there was trouble with the electron deficient anilines. They didn't really want to go. I sent a reply back saying maybe you could try coupling to something like amantadine, some sort of isostereophenol.
Well, so someone who's familiar with the synthesis required, maybe I should just try and go to that because I'm gonna.
Yeah, it should be somewhere under chemistry.
That's the oxide chloride one, CDI. Okay, all the different kinds of couplings, right? And then, okay, all right. So it is, all right. So
And
it at the moment it's one step, right?
yes, yeah. And you've got the York ELN down there as well. Though I haven't had a look at that myself, so I don't know
Right,
how easy it
right.
is to see what they're up to in a glance.
Okay, but it is one step. So the typically speaking, do people know if I mean, this is laid out like a really nice recipe. For the purification. So I'm just wondering about the use of these kinds of processes in schools or with undergrads and stuff. Is this something which we ought to be suggesting to, for example, schools as something that's realistic? Because it's a mix and then it's a workup and then it's some kind of purification. Is
And
it that easy?
I guess I would say that it's theoretically easy, but I'm going to just quickly see if I can find it on the UCID lab archives. I think basically they found that the HBTU coupling worked the best,
Mhm.
but it's quite expensive and a skin sensitizer, so I wouldn't recommend it for schools. even in the way that we do it with the Breaking Good workshops, where it's much more supervised. I think some of the other methods did work okay. And, you know, given that if you're doing it with schools and they're making 5 grams or 2 grams, whatever, it's fine if the yields are terrible. There's definitely potential, but yeah, I can't remember which of them worked better or worse off the top of my head.
And the purifications are just columns, are they? They're not crystallizations, I'm guessing.
No, they were just columns, and the students didn't do them at UCID. That was another researcher, basically.
Sure.
Yeah, I think they were quite messy. I didn't do the purification myself. I just chatted to the guy who was doing it, but he wasn't thrilled with them.
Right.
But it's hard to know how much of that's just the students.
Yeah. I mean, in terms of school synthesis, you know, this is pretty much ideal that you get a couple of things in and mix them together, some reagents, and work up, maybe do a TLC or something, and then try and purify. But yes, it has to be reasonably robust chemistry in the sense it needs to some extent work as intended, and B, if there's columns necessary, then it's likely that a university local to the school would have to step in and help out. But in terms of being one-step process, that's pretty much what you want for a school project, I think.
Yeah, I don't know with the columns, because we just don't bother doing anything like that in the first year labs. It's possible that they would be able to do it, you know, maybe not in a school, but certainly in an undergrad lab situation. But yeah, I guess just depends. Sorry, the lab archives is doing the 2-factor authentication and being difficult.
Okay. All right. But yes, I mean, in terms of you know, working with Whitgift, for example, and other schools, I mean, it does seem this would be a really good thing to do. We, you know, in the aminothiazole stuff, I mean, the chemistry is also really good, but there is this issue that it's a at least two-step process. And so sometimes, you know, the procedure was completed and we weren't really sure which bit of it was a problem. Whereas here with a single step, it's pretty nice.
Yeah, definitely.
And there's also the esterification thing that Bernard just mentioned, you know, as a possibility for adding in some interesting complexity.
Yeah, so those compounds that were ordered were very simple ones. They were so if they come back potent, that would be amazing, because one of them was something called benodinil, which is literally the parent compound. It's salicylanolide, so it's just the diphenylamide with an iodine on it. So if that comes back potent, maybe we can make like the 2,5-dichloro version of that or something, and that would be probably pretty easy. So those are very simplified analogs in the issue 13.
Okay.
That's why I picked them.
All right, great. Okay, and then so I'll just also on the landing page and the wiki. So I haven't checked it myself, so I ran out of time. Does the project page make more sense now, do we think? Hopefully. In the sense of
I updated it, so I think it makes sense, but it would be
okay,
I don't know
good.
if anyone else has looked at it.
I proofread it.
Okay. Oh, this is this thing. Right. Cool. Okay, great. And then
yeah,
all
I
right.
just wrote the wiki home is kind of just a like a default, you know, could be on any of the series.
Right.
And then I tried to follow what we have on the series 2 wiki so that it's, you know, familiar. Yeah, so we have the origin. I think my About MycetOS page basically just links to other pages that we already have written elsewhere. Yeah, goes to our repository. And yeah, so I don't think I missed anything crucial, which basically there were a couple of compounds from various different screenings of drug libraries. Then the compounds donated by Dr. Melander at the Uni of Notre Dame, and then we're making compounds for students at the moment.
Mhm. Yeah, perfect. Okay, all good. Yeah, looks good. Just to give, newbies a basic sense of where this has all come from.
Yeah, no, it was good. I think there's some data that I should add to that Anthony found for me, because like the data that's on there, those compounds have been tested against a few different strains of mycetoma. We don't have a full set of data for all of the strains, but there should be data for MM55 for all of them. I just need to
Right.
go and add it.
Okay. All right. Great. All right. Fantastic. That's great. Great. Thanks for doing that. That's really much better.
Yeah, also the compounds that were being ordered are not in the SAR tables, but I mean, Dunya said they haven't really, only one came out of the four, so
When they
Do you
come
have
in.
the link to like the page that they're on just handy?
it's that issue 13 right there, that commercially sourced compounds. You can
Okay,
click
yeah,
that.
I'll just I
That's
can
issue
add those in.
13. Yeah. Yeah, I picked those out because if they're pretty simplified, so it would be amazing if the Nodonil came back potent because that would be extremely easy to make analogs of.
Yeah, fingers crossed. And do you remember which one it is that arrived?
Yeah, I think it was the just salicylate anhydride. Let me check my mail. It
I'm
has
guessing
been,
that they are, well, that there's more than one that has that name.
yeah, it's called salicylate anhydride.
Yeah. God
The
damn. Okay, so
that's
that's S0007.
yeah, that's the parent compound, so that's basically without all the substitutions on it. It's just a hydroxyl group on the diarylamide skeleton.
Well, that'd be good to see as a, bringing it back to basics.
Okay, fantastic. Anything else on Series 7? Because we have no dedicated chemistry on this now. So, being able to make the chemistry straightforward would greatly enable us to access compounds from more sources. I mean, I know Peter and Shane have students on these, but I mean, there's only so much that can be done. We can broaden that out by making the chemistry very simple. That would enable us to get more compounds.
A lot of these can be purchased. There's a company called MQL that sells them, but their
Yeah.
pricing is a little bit concerning. It can vary a
Well,
lot.
yeah, so
Pricing.
if they are simply available, if they're already available, they should be there for about, $60 or something. If they have to be made, it'll be like 100 and something. So absolutely you can do that. You just got to find money for it, or integrate this into a lab class and generate it that way through something educational. Those are the two ways through. If we had a grant for this, then it would be a mixture of both.
I mean, I don't want to speak too much for Shane, but I suspect that if there's, one or two compounds that we could purchase that are, helpful for the SAR, they could probably figure out how to put that into the education, the lab program budget.
Yeah.
So, but yeah, we'll just have to be very selective about which ones.
Yeah, yeah. I mean, if you're going to do chemistry on a series like this, then buying molecules is definitely the fastest way to go. Provided you've got the resource for it, as always. Okay, great. Thank you for all those updates. I'm going to transition to the any other business. So and if you remember this, we were interested in trying new targets for to kickstart new projects by by determining some understudied or promising looking proteins that we could recombinantly make and then put into the SGC screening pipeline, and came up with these two, SID-A and MKK2, and have been trying to source samples of them from mycetomatis, but also from Aspergillus fumigatus. And I had an update just earlier today from Jelena, who was working in my lab, who has been working on getting a sample of SID-A from Aspergillus. And the short update is that the SID-A is expressing really nicely. So this is the gel for SID-A in the two different broths here, and it's expressing really well and really cleanly. And so in progress is a scale up of that to generate a couple of milligrams or so that is his tagged and suitable for putting into the screen, the ASMS screen. The MKK2 is being more tricky, but isn't dead yet. But that's looking promising. So hopefully when time permits, because there's lots of other things on Jelena's plate, they will be able to scale it up and ship it off for screening versus the new SGC ASMS library, which is this 20,000 racemate library, which will be great, because then if we get hits from that, then we can confirm those and try and find a bit of resource again to buy some molecules around that hit to see if we can get something which really holds up. And if we can, then that kickstarts a whole new program versus that target. So that's really good. I wondered whether or not the Erasmus MC team had any updates related to the mycetomatis version of those proteins.
Saskia, do you want to give the updates?
Yeah, so well, we don't really have a lot of updates. We do have a master student now that started on this project. So he started, well, in the lab beginning this month. So we will start this week with the induction of expression in E. coli. So hopefully next time we can share a little bit more.
So the plasmids are in. We did do 3 proteins of Madurella mycetomatis, so MKK2, SID-A, and also PRA1. The zincophore we discovered before. And yeah, so let's hope that at least SID-A also expresses nicely, like in your case, but maybe also PRA1. So we decided to go for 3 targets.
Right. Okay. Fantastic. All right. Fingers crossed. Okay. Yeah, there was just a residual question that I saw that Mars thesis defense happened, and I just wondered if we'd link to that somewhere, because I think we had the live stream link, did we, last time? And I wondered if we should just put that somewhere if we haven't already, as outputs from MycetOS.
Yeah, I put it where I put Wilson's thesis, so it is
Right.
But it's only Wilson's thesis there, so there might be several spots on GitHub where we host these things.
Yeah, we do have 2 different pages with them. So I copied what Wendy had put up and put it into the other place, but we should maybe another time discuss where to keep things. In the other place we have Hung and Dimitri, as well as Wilson.
Right. There was it's in the talks, meetings, publicity repo. Is that what it is?
Yeah, there I put it. But I couldn't find Hang and Dimitri there, so.
Okay. No. Oh, papers and theses. There's a general start here, isn't there?
Yes.
That's got. Right.
I think this is
in
probably
there. There's the whole list there.
Yeah.
Well, that's pretty. Yeah. I mean, as long as we're consistent and we link to it, then
possibly we should yeah, talk about it another time when there's, when it's not almost lunchtime.
Yeah.
But
Yeah, and okay. But
um,
yeah, that's good though, that is here.
we have a lot of repos that we don't really use. And I mean, I'd be curious to know what other people think, but I think overall it would perhaps be better to have fewer active repositories with more stuff in them, because it's a bit,
Mhm.
It's hard to remember, oh, wait, there is that other repository over in this other place. Let me go look there as well.
Yeah.
But we'll have to
Yeah.
kind of sit down and look at them all in one go.
Yeah. Yeah. But I mean, it is amazing that's a pretty impressive page, actually, which is the desired effect, is that this is an open source project with very little direct funding to it, that we are getting together a bunch of things. And there are a bunch of papers here and 4 PhDs. I mean, that's pretty good, I think.
I have a
You know, we
question.
should, let's also pat on the back. Yeah. But
Yes, as you can see, we have published quite a few papers now with some very promising compounds, and I want to ask if there are plans about some of the most promising compounds to advance to more advanced animal tests, like tests with infection models in mice or rats or something like that, to get forward with the development of these compounds. And
Well,
maybe
there are
to achieve one time that we can reach clinical trials even then.
Yeah, so currently we have one grant from the Leo Foundation to develop a mouse model. So that means that for now, the Galeria is the best we can do. So we cannot go further because there is no good model at this moment in time.
Yeah, maybe we can write a post on LinkedIn that we are searching for suitable partners in this regard.
Yeah, but that's the problem. There are no mycetoma mouse models.
Ah, okay.
Wasn't there previously a mycetoma mouse model?
Yes, we had. It was an
Yeah.
intraperitoneal model, but it is not that very good because you need to open up the mice after 20 days and check if there are grains visually in the interperitoneal region. So you cannot follow over time very well. Mycetoma is not a lethal disease, so we are simply working on a better model. So when that's up and running, then we could consider this, but not before that, I guess.
Yeah, because at the moment we are stuck a little in the preclinical stage, and yeah, maybe we can advance then to more sophisticated tests than in vivo tests, and get some information how to reach even clinical trials then in the end to get a new drug for this disease.
Yeah, but in those cases, then funding is really essential because
Yes.
running a Galeria model is one thing, but running a mouse model, that's a whole different financial thing.
I think mice or vertebrates are crucial to advance in this regard.
Do we have like a list somewhere of compounds that would be our first to go into a mouse model if we had a mouse model suddenly
So
ready to go?
I think the last review we did on all the things we found was the paper from Wilson, number 6 on this screen.
Yeah.
Yeah, so maybe when we publish a few more of the papers, that we should simply update this review.
yeah, it would be it'd be very interesting to get the perspective from our DNDi partners on this also, if we were to compile a list of that kind, whether it is seen as being a good thing if we can try and make sure that any molecule we try and progress has a new mechanism of action rather than one that goes against the well-worn target. It'd be nice to know their view on that. But I agree, it might be nice to have a list somewhere here that summarizes the best compounds that we've identified to date that we would be interested in progressing were such a model to be available. Yeah, it's a good point. Good luck with the project there,
Yeah,
Wendy.
because another issue with these mice tests then or rat tests will be that you need more amounts of the drug. Yeah, that you need 1 gram or 2 grams of the drug to feed all the mice. Then
Sure.
not just a few milligrams that are now useful for the larvae tests.
Yeah, plus the previous mouse model was also a very lengthy mouse model. So you're thinking about at least 2 to 4 weeks treatment schedules. So that's a lot of compounds you need usually.
We're on the hour, so just 2 very quick other things. For the Gmail account, I mean, I know how to log into that. If Max or somebody else wants to share that, absolutely fine. We can do that offline. And this thing with a lab notebook, I'm currently negotiating extensions with Lab Archives. Hopefully that will happen before that deadline comes into play. But don't worry, we are doing everything we can to make sure we're not going to lose anything, and that we carry on our subscription, at least for the moment. There's a slight difference because Lab Archives has been acquired by somebody, and the terms are slightly different, so it's a bit complicated.
Just quickly on papers as well.
So just quickly on that. So students are still using Lab Archives, right? Through us, like Whitgift and people like that, right? They're still using, and Peter's still using ours, right? I think.
Yeah, I believe so.
Yeah, right. Okay. All right, fine. Sorry, I interrupted.
Yeah, I just wanted to ask, I know that Dimitri talks about ketoxenes in his PhD, but is there a paper on that, or has it not made it into?
No, there's no paper. So we've got the data and we've got write-up, in PhD. There's an issue on that repository which lists all the things which need to happen next.
Right, before paper.
We're stuck there.
Yeah, okay. Just wanted to check I wasn't missing one.
Yeah. All right. Any other rapid business, or are we, because we're out of time? Okay. Thanks for coming, everybody. Nice to see you all.
See you next month.
Yeah, see you next time.
Bye.